The NADPH oxidase is a membrane-bound enzyme complex that faces the extracellular space. It can be found in the plasma membrane as well as in the membranes of phagosomes used by neutrophil white blood cells to engulf microorganisms. Isoforms are designated NOx1, NOx2, NOx3, and NOx4. Subunits: it is made up of 6 subunits. These subunits are aroguanosine triphosphatase, usually RAC1 or REC2, 5-PHOX units, GP91-PHOX, NOx2, P22-PHOX, P40-PHOX, P47-PHOX, P67-PHOX. Function: under normal circumstances, the complex is latent in neutrophils and is activated to assemble in the membranes during respiratory burst. NADPH oxidase generates superoxide by transferring electrons from NADPH inside the cell across the membrane and coupling these to molecular oxygen to produce superoxide anion, a reactive free radical. Superoxide can be produced in phagosomes, which contain ingested bacteria and fungi, or it can be produced outside of the cell. In a phagosome, superoxide can spontaneously form hydrogen peroxide that will undergo further reactions to generate reactive oxygen species. Superoxide kills bacteria and fungi by mechanisms that are not yet fully understood, but may inactivate critical metabolic enzymes, initiate lipid peroxidation, and liberate redox active ion, which allows the generation of indiscriminate oxidants such as the hydroxyl radical. It is presumed that superoxide kills bacteria directly, as the virulence of many pathogens is dramatically attenuated when their superoxide dismutase genes are deleted. However, downstream products of superoxide also include hydrogen peroxide and hypochlorous acid, the reactive agent in bleach. Role in pathology: NADPH oxidase is a major cause of atherosclerosis, and NADPH oxidase inhibitors may reverse atherosclerosis. Atherosclerosis is caused by the accumulation of macrophages containing cholesterol in artery walls. NADPH oxidase produces ROS. These ROS activate an enzyme that makes the macrophages adhere to the artery wall. This process is counterbalanced by NADPH oxidase inhibitors, and by antioxidants. An imbalance in favor of ROS produces atherosclerosis. In vitro studies have found that the NADPH oxidase inhibitors apocynin and diphenyliodonium, along with the antioxidants N-acetylcysteine and resveratrol, depolymerized the actin, broke the adhesions, and allowed foam cells to migrate out of the intima. Mutations in the NADPH oxidase subunit genes cause several chronic granulomatous diseases, such as X-linked chronic granulomatous disease, autosomal recessive cytochrome B negative CGD, autosomal recessive cytochrome B positive CGD type I, autosomal recessive cytochrome B positive CGD type II. In these diseases, cells have a low capacity for phagocytosis, and persistent bacterial infections occur. Areas of infected cells are common: granulomas. A similar disorder called neutrophil immunodeficiency syndrome is linked to a mutation in the RAC2, also a part of the complex. One study suggests a role for NADPH oxidase in ketamine-induced loss of neuronal parvalbumin and GAD67 expression. Similar loss is observed in schizophrenia, and the results may point at the NADPH oxidase as a possible player in the pathophysiology of the disease. Nitroblue tetrazolium is used in a diagnostic test, in particular, for chronic granulomatous disease, a disease in which there is a defect in NADPH oxidase. Therefore, the phagocyte is unable to make the reactive oxygen species or radicals required for bacterial killing, resulting in bacteria thriving within the phagocyte. The higher the blue score, the better the cell is at producing reactive oxygen species. Chemical reaction: NADPH plus 2O2 NADP plus 2O2 plus H plus. Inhibition: NADPH oxidase can be inhibited by apocynin and DPI. Apocynin prevents the assembly of the NADPH oxidase subunits. Using opocinin to inhibit NADPH oxidase may have clinical benefits in the treatment of influenza as it lessens influenza-induced lung inflammation in mice in vivo. See also, NOx1, NOx2, NOx3, NOx4. References. External links: NADPH oxidase at the U.S. National Library of Medicine. Medical subject headings: EC 1.6.3.1